my pleasure to introduce a very, very dear friend of mine who we are so blessed to have here in Indiana. I met him 8 years ago, and he has changed the face of research here in the state of Indiana. And, you know, he's one of those guys, and I'm going to call you Hoosier now. I don't know if you're going to accept it or not. But you know that, like, you're talking to this world-renowned thought leader that everybody on the planet knows who he is. And he's just Bruce, my friend. So I would like to introduce Bruce Lamb from the Stark Institute.
Morning, everybody. It's really great to be here. I want to start by thanking BioCrossroads for hosting this event. We were talking earlier about, there was BioCrossroads events, I think it was 5 or 6 years ago on Alzheimer's. And I think it was really important to just think about where we are today compared to then. And I think it's just remarkable. And again, you heard some of that this morning, and I think you'll continue to hear that through the rest of the day. So I wanted to start by giving an introduction. So I'm hoping in my talk to sort of basically introduce sort of the next panels. So we'll all have some of the same understandings to sort of really get into deep discussions of the panel. So what I hope to do is on the slide here is to really talk about how Indiana, I think, really is leading the way in some of the new era of treatments, as it's being called. And so that starts with a history, just very brief, of Alzheimer's disease research, a history of therapeutic development, talk a little bit about the new disease-modifying treatments that you've been hearing about. Also talk about how IU and IU School of Medicine in particular has really been leading the charge in terms of some of the research, the national and international research programs focused on Alzheimer's disease. Talk about a little bit about the recent sort of advances in therapeutic development and implementation challenges for the health systems. We heard a little bit about that in this last panel, and I think that's just something I want to touch down on. Talk a little bit about the IU Health Brain Health Program, which is, again, bringing these treatments forward into patients for the first time. And so just wanted to mention that. And then the last thing, just to touch down on also, because I think it's going to be important for the panels, is to talk about health disparities and access to treatments and some of the struggles we're going to have in that area that I think are important to have a discussion about. So the history. So the history of Alzheimer's disease really starts in the early 1900s. The Bavarian neuropathologist Alois Alzheimer, who's on the left side of this slide, and a patient who he saw, August Dieter, who's on the right side of the slide. And he sort of presented her case on November 3rd in 1906. And being a neuropathologist, he looked in her brain when she died and observed sort of a unique neuropathology. And you've already heard some about this today, but just want to sort of reinforce it. So the first two pathologies that he observed, and actually if you look on the right side of this slide, you actually see drawings. So some of those are drawings of Alois Alzheimer. In the early 1900s, he did not have a camera. He was drawing what he saw. And then you actually see some of the actual sections which were found many decades later actually in his laboratory. So they actually were able to find these exact sections from August Dieter's brain in the laboratory. But what he observed were two different hallmark pathologies which have really defined the disease for the coming decades, or really coming century. And that is the presence of what are called senile plaques. And these are deposits, extracellular deposits of a little peptide called beta amyloid. And again, we've been hearing some already about this. And the second is our neurofibrillary tangles, which are those flame-like structures that Alois Alzheimer, you sort of see those flame structures that he drew. And this is intracellular accumulation of another protein called tau, again, which you've also heard a little bit already. Importantly, the neurofibrillary tangles are observed in a variety of other neurodegenerative diseases that involve dementia. And again, you've been talking also this morning about some of the complexity. But it's really sort of these two hallmark pathologies since the time of Alois Alzheimer's that sort of has defined the disease entity as we know it. And importantly also, as Phyllis, I think, mentioned earlier, up until very, very recently, really we required a post-mortem diagnosis in order to really diagnose the disease. And that obviously is a real problem. And it's really only been very recently. So if we now look at some of the history mentioned in early 1900s, Alois Alzheimer first described the disease. There was a hypothesis about cholinergic deficits, which are the basis actually of our current previous class of FDA-approved treatments, has actually improved that cholinergic working of the brain. Then the amyloid hypothesis was established, which really puts at the basis that amyloid is at sort of the central part of the disease, and this was both based on genetics and biochemistry, which really suggested this was the case. And it was really, I think, a unifying hypothesis for the field. The amyloid precursor protein, which is the precursor for this little amyloid peptide, was cloned. The first animal models were generated in the mid-1990s. And I think what I really want to focus on next, again, we had a couple of approval of these drugs that, again, help sort of nervous system function, but they are not disease modifying. They're more symptomatic, really, treatments. And then really what I want to point to is in 2012, when we had sort of the first approval by the FDA of now an imaging agent that could image amyloid live in people. And again, Lilly sort of really, again, and I want to highlight Indiana sort of throughout here, and Lilly was really central in some of those efforts, and Amyvid, as its name, was developed and then approved by the FDA for amyloid imaging, and that was only in 2012. Again, just to give you an idea of how fast we have come. And then a few years later, the tau PET, so being able to image tau in living patients was also developed. So that also now we have in our armamentarium of sort of basically diagnostic tools. So again, that's all happened in the last decade or less. And then we'll hear later a bit on, I'll talk a little bit more about the other biomarkers, which I think are revolutionizing or going to revolutionize diagnostics as well. And out of all of this came in the 2000s multiple trials targeting both beta amyloid and tau. Again, up until very recently, it has been a very difficult field in a lot of ways. There's been a lot of disappointments. And again, just a few years ago, I remember coming back from conferences feeling, are we ever going to get there, to have something? And I would sort of say we have definitely arrived. There's also lots of things that I think have made this complicated. So the timing of treatment, how do we figure out when to treat the disease? The heterogeneity, we heard a little bit already about this, and I think you're going to continue to hear about this through the panels. The disease is very heterogeneous. Yes, there's amyloid and tau, but often, especially in late onset disease, you have lots of other comorbidities and coexisting pathologies in the brain existing at the same time. So how do we think about that with regard to treatments? Again, the right target at the right time. How do we, again, are we working with the right targets? And then also biomarkers. How do we really move forward in understanding all of this if we don't have really good translational biomarkers? So in terms of the therapeutic development, as I mentioned, we had a variety of anti-amyloid strategies, anti-tau strategies. The anti-amyloid therapies included antibodies targeting amyloid so it can remove it from the brain, secretase inhibitors that prevent the formation of amyloid in the first place, things that prevent amyloid aggregation within the brain. And similarly for anti-tau, there's been antibody inhibitors for kinases which phosphorylate tau, which are thought to be one of the catalytic aspects of the development of tau pathology, as well as aggregation inhibitors. But up until 2021, again, none of these had been successful in phase 3 clinical trials. And this just shows, again, some of the clinical development, where, again, we had early drugs that were really aimed at sort of improving neuronal function, as I would call it, sort of early on, and symptoms. And it wasn't really until 2021 when aducanumab, developed by Biogen, was approved by the FDA and in the accelerated approval pathway. And then Eisai and Biogen got lecanemab approved first by the FDA accelerated pathway in early parts of this year. And then over July got FDA traditional approval, and CMS actually now agreed to pay for the drug. So that, again, it was a huge development, again, over a very short period of time that we're talking about. And then finally, Lilly has announced their phase 3 results for donanemab, which is another monoclonal antibody against beta amyloid. And they announced their results at AAIC over the summer in Amsterdam, and have already submitted for approval at the FDA and likely hear something even by the end of this calendar year. So again, just incredibly exciting times, a lot of things going on sort of all simultaneously. So I wanted to step back a little bit and talk a little bit about what these drugs do, because I think it's important probably for some of the conversations we're going to have. So the first thing I want to just point out with this slide was that there's a lot of different forms of beta amyloid. It's a 40 amino acid peptide, but once it's generated, it can exist in a lot of other forms, sort of within the brain. And this includes what we call monomers, which are just these little peptides on their own when they're first generated. But it includes what we call A-beta oligomers, which is groups of smaller numbers of these monomers, what are called protofibrils, which are even larger accumulation aggregates of fibrils, the A-beta fibrils themselves, and then finally the amyloid plaque, which is the thing, again, that's seen by PET imaging when we image it. And importantly to think about is that all of these are sort of in equilibrium with one another within the brain. And so it's important thinking about when we start thinking about these therapeutics, what are we targeting and what impact is that going to have? And so if we now look at the different drugs that have been developed, we know that aducanumab, again developed by Biogen, largely sees the amyloid plaque and to a lesser extent the oligomers and the fibrils. Lecanemab, developed by Eisai in collaboration with Biogen, preferentially sees the protofibrils, which is another form, although it does see both the A-beta fibrils and oligomers. Lilly's drug, donanemab, monoclonal antibody, really sees these pure amyloid plaques, the sort of end stage of the aggregation of beta amyloid. And then Lilly also had another drug which failed in phase 3 clinical trials, which is solanezumab, which is monoclonal antibody against this soluble form of beta amyloid. And then finally, and it's again relevant, I think, to our Indiana story, is this drug ACU-193, which is another monoclonal antibody which selectively sees A-beta oligomers, developed by Acumen, which is sort of an Indiana company here in town. So again, you can sort of see first two things. I mean, I think there's a lot of different efforts underway trying to target different forms of amyloid, and that Indiana is certainly playing a key role in a lot of this work that's going on. And so now to get back to sort of how these drugs have worked. So this graph, and I got this from the Alzheimer's Association through Jeff Cummings, who sort of helped generate this. And so on this graph, on the x-axis, this is sort of removal of amyloid. And so there's more removal on the left side of the graph, less removal on the right side of the graph. And then the y-axis is actually cognitive benefit, which is called the CDR sum of boxes, which is a cognitive assessment tool. And what you sort of see here is improvement in cognition going down on the y-axis and no improvement moving up. And what you can sort of see is there's a relationship between how much amyloid can be removed and the cognitive benefit. And again, amyloid being defined as this that's seen by the PET imaging, which is those amyloid plaques itself. And I think this is really an important observation, which again has been repeated now in a lot of different clinical trials, that you can sort of see that those trials that remove amyloid and remove it fairly quickly are the ones where we're beginning to see a cognitive benefit. And I think that's sort of really the take-home message thus far, I think, in a lot of those trials. And so now to look at just a little bit of data for these trials. So this is donanemab, but again, similar data, actually, if you look at the lecanemab trial as well. So donanemab definitely clears amyloid from the brain. There's no question about it. And it does so relatively quickly here. So you can sort of see now the percentage of participants in this graph that are actually basically clearing amyloid by these different time points. So at 24 weeks, you can see almost 30%. By 52 weeks of treatment, 66%, and at 76 weeks, 76%. So it clears amyloid in a lot of patients and does it relatively quickly. And that's, I think, and then in addition, the Lilly trial actually allowed then those individuals who had cleared amyloid would actually go off the therapies as well, because that amyloid plaque is no longer there. So again, I think this is a new era. We still don't know a lot what's going to happen to those people as they now have amyloid removed and continue to go on. But importantly, and that's shown in this slide, there is a cognitive benefit. So these are in two different cognitive measures on the left and right, iADRS and CDR sum of boxes on the right. And basically what you sort of see in the population as a whole, either a 22% or a 29% slowing of cognitive decline in the total phase 3 result. And again, this is the first time ever, and I want to emphasize this has been seen, given lecanemab in this study and aducanumab in one arm of that trial as well. But I do want to also come back to, I think there's a number of things we also need to think about. First, looking at the graphs, you can sort of see, yes, it slows cognitive decline. It does not stop it. It does not reverse it. And so that's one thing I think we all have to keep in the back of our minds. And so one of the things we need to think about in considerations is this is targeting early stage of disease. So these are people either with mild Alzheimer's disease or what's called mild cognitive impairment, which is very early stages of disease. So this would not be indicated for anybody who is at the moderate or severe stages of Alzheimer's disease. And interestingly, and I think Phyllis mentioned this, data presented at CTAD 2 weeks ago actually showed that individuals in this trial, and donanemab and lecanemab trial, who are at an earlier stage, actually there's a bigger clinical benefit. So I think that's a really important take home that, again, we need to think about as we're moving forward with these trials as well as others, that we need to start as early as we possibly can, and all the issues that go alongside of that. These are also infusions. So again, you have to go into the hospital to an infusion center and get the drugs. But again, presented at CTAD, there's now a subcutaneous formulation for, at least for lecanemab, which suggests it also removes amyloid from the brain. So again, that would then allow injections to occur as opposed to an infusion. There's also side effects of these medications. There's what's called amyloid-related imaging abnormalities, which is thought to be sort of an inflammation in the brain. And it can be serious. Typically it's not, but it can be quite serious. And so that's, again, something also we need to understand a lot more about, and hopefully will be part of the panel discussions. And then I think where the field is moving, and it's already moving into this, is now combination therapies. We're immediately entering the age of combination therapies. How do we move together with these anti-amyloid therapies plus other new therapeutics moving forward? So just now to talk a little bit about NIH funding and the research here in IU. So NIH funding, thanks to, again, a lot of the work by the Alzheimer's Association on the advocacy side, has completely changed federal funding for Alzheimer's disease research in the United States. So since 2014, there's been an over 700% increase in federal research funding from the NIH. And this has completely transformed, I think, what we've been able to do, and I think why we're at the point where we are right now in the field. And this has included a number of large initiatives which are shown in these hexagons here, which are different translationally focused programs trying to identify the next therapeutics. I've talked about amyloid and tau, but what are other therapeutics that we should be thinking about behind that? And that's what these big programs are. And IU School of Medicine has really been at the center of a number of these very large programs, and we are leading a number of them. So this includes, and again, you don't need to go through all of them, but National Centralized Repository for Alzheimer's Disease, NCRAD. All the samples from all across the country come here to Indiana. So all the Alzheimer's centers send their samples here to Indianapolis. The Longitudinal Early Onset Alzheimer's Disease Study, which is a longitudinal international study now of this unique early onset Alzheimer's disease. We're also part of this CLEAR-AD, which is a program to identify biomarkers, new biomarkers for Alzheimer's disease. Model-AD, which is an animal model development and also preclinical therapeutic testing program that I help lead as one of the national programs. TREAT-AD, which is a drug discovery center, one of two funded in the US, led by Alan Paukowitz and myself, as well as a large team of investigators. And then, again, there's many different institutions that are involved here as well. IU School of Medicine, for sure, Stark Neurosciences Research Institute, where I am, but also the Regenstrief Institute and the Indiana Biosciences Research Institute are all engaged in these processes. And so we have funding across the entire spectrum, everything from the most basic science to sort of clinical programs, and how do we get these treatments out into the communities. And then finally, so we have this whole, I think, research going on in every aspect of the translational research program. It was mentioned that we're fourth in funding from the National Institute on Aging, a major funding for Alzheimer's disease in 2021. And then out of the Treat ADC Center, as well as through our efforts as Model AD and Stark, we've also launched a small biotech called Monument Biosenses that you're going to be hearing more about today as well. And then finally, and I put it in here because I wanted to just announce that this is the most recent one, this ADACC, which is an effort led by Nicole Fowler, as well as Jeff Daig is involved and others at IU, which is really an Alzheimer's diagnosis in adults with other comorbid conditions. How do we start thinking about, again, this more complexity of the disease, and how do we look at biomarkers in that context? So again, that's another new exciting one. So I just wanted to emphasize quickly, and I guess this was mentioned already, while amyloid and tau have been a major focus of therapeutic developments, this is out of an annual paper that Jeff Cummings put out about therapeutic development for Alzheimer's disease. And on the right, you can sort of see while there are a lot of trials going on for amyloid tau, sort of at the bottom there on that right-hand panel, there's lots of work going on in lots of other areas, inflammation, neurotransmitters, oxidative stress, et cetera, a number of different biologies that are non-amyloid, non-tau, and I think those are only increasing as we move forward. The other thing I want to just touch down on was biomarkers. And just so for definition, a biomarker, which is short for biological marker, could be considered a measurable substance or a characteristic that indicates the presence and or progression of the disease. So biomarkers can be a variety of different things. They could include proteins, genes, antibodies, imaging, digital biomarkers. There's a whole number of different things that could be considered in that mix. And importantly, biomarkers can be used to identify risk for disease, to measure disease, and also measure efficacy of treatments. And that's really, I think, this area has exploded the most over the last 5 years, I would say. So we now have, as I told you about before, PET imaging, where we can image in living individuals, amyloid and tau. We now have a number of biomarkers in the cerebral spinal fluid. So taking a lumbar puncture, we can identify biomarkers in both amyloid and tau, which clearly link to disease and can help us develop diagnostics for disease. And finally, the last group that's sort of been the most latest, and I think perhaps the most exciting addition to this, is plasma biomarkers. So we now have evidence that there are plasma biomarkers, including beta amyloid and tau, that for the first time are allowing us to sort of determine whether individuals at least are at risk for developing Alzheimer's disease. And again, this has the potential, I think, to really transform the field because plasma biomarkers are a lot cheaper first than doing PET imaging on every individual and can be brought out into the community and ultimately even into primary care. So this just shows some of the blood-based biomarkers. I mentioned what we call amyloid beta peptide ratio, which are these two disease-relevant peptides, 42 and 40. Phosphorylated tau, one of these assays developed by Jeff Daig while he was at Lilly. And then finally, neurofilament light chain, which looks at neurodegeneration. And this is based upon this NIA Alzheimer's Association 2018 research framework, which suggested that ATN, sort of the amyloid, tau, and neurodegeneration are the things that help us define the disease. And importantly, in biomarker work, I sort of say we have so much work going on within Indiana. We have Roche Diagnostics, which is here in town, has a major program focused on Alzheimer's disease biomarkers. Lilly has similarly, as I mentioned, some of the imaging agents they brought forward, but they're also working, I know, hard on the fluid biomarker programs as well. I mentioned NICRAD, this program at IU School of Medicine, and in the Stark Neurosciences Research Institute, we have a biomarker discovery lab led by Jeff Daig. So this just shows, again, where we are in terms of the development of these. So the markers here shown in black are basically now sort of approved, amyloid PET, tau PET, and up until now we've been able to use MRI or FDG PET for looking at neurodegeneration. Those that are in purple right now are only research only, but again, I think it's only a matter of time because before these are going to enter sort of into the clinical space as well. So stay tuned, I think that's going to happen. And so finally, I wanted to touch base on one of the other issues, which is, okay, we're in this era of treatment, and you've heard a little bit about this on the first panel. How do we now think about this in terms of implementing these treatments, given the challenges we have in healthcare systems in the United States? So there's a number of issues. Detection, so this is both looking in primary care, pipelines and protocols, and you heard a little bit from Jared about some of our findings from the Davos-funded initiative. Imaging prior authorization, referral pathways for advanced diagnostics, all of those things are things, again, we need to figure out. Diagnosis, resources for biomarker testing and genetic testing, as well as counseling, drug administration, pathway registry, and thanks to the Alzheimer's Association, they've launched Alzheimer's Network, which is a patient registry, but to basically develop a nationwide registry for patients. Again, you can sort of see some of the other areas that I think we all are going to struggle with as a health system. And then care planning and care management. And again, there's some now nationwide efforts. And again, some of those are led out of the Regan Street Institute and IU School of Medicine, looking at how do we sort of launch these into the different communities. So this, again, just goes back to some of the challenges. Again, primary care, specialist shortages, referral bottlenecks. Again, you can sort of see the entire list of things that we've been considering. And again, there's a lot of work going on trying to understand this. So you heard about the Davos initiative together with IU Health and the Regan Street Institute to really sort of try to understand what the barriers are in primary care. The Alzheimer's Association similarly is engaging health systems trying to identify how do we get around a lot of these barriers in health systems. But again, it's a major challenge doing this. So just as a way of an example, so Donna Wilcox and Liana Pasalova have led a team, including Jared Brosh, who you heard earlier, Jeff Daig, and others, to develop what we're calling an Indiana University Health Brain Health Program. And you already heard about the Brain Health Navigator, again, an individual who will be there who can help navigate individuals as they move through this process, cognitive battery, MRI to check for microbleeds or microinfarcts to suggest that maybe they're not the ideal subjects for these infusions, PET or CSF for amyloid positivity, potential ApoE genotyping, and we come back to this. This is a genetic risk for Alzheimer's disease. And then a pre-authorization team, and then finally initiating treatment. And the idea is, too, you need to have this process all developed out and quickly so you can get patients in as fast as possible because they can move out of the stage at which they are eligible for treatment. And I think that's a really key point. So this just shows, again, so we're in the phase 1 rollout timeline. You can sort of see here we launched sort of in mid-September of this year. The first treatments at Indiana University Health and our continuing enrollment as we move forward. So far, this is as of October 27th. I don't know if there's more since then, but there were 13 total patients. There were 5 patients waiting on insurance approval. There's 1 patient starting that week, and 6 more patients completing screening process to start treatment in the next 2 weeks. So again, this gives you really up to date. We are right now just experiencing this for the first time, sort of in real life situations. And then the final thing I just want to touch down on, because I think it's important, and we haven't really discussed it too much yet, but it's a really critical thing, is disparities. Both in Alzheimer's prevalence clinical trials, as well as potential access to these new treatments. So older Black and Hispanic Americans are disproportionately affected by Alzheimer's disease risk. So approximately 2 times and or 1.5 times more than Caucasian populations overall. Ethno-racial groups have historically been underrepresented in clinical studies for sure across decades. And importantly, less than 5% of all clinical trial participants are from these underrepresented groups. And so, again, this is a huge issue, I think, for us as a community. And then also, I think there's concerns that in addition to those concerns we already have, now, how about access to these new treatments, to these communities? And again, there's some barriers there, as you heard about. We have to be in academic medical health centers that have these capabilities of infusion centers and MRI and PET imaging, all the rest. So how do we think about this? How do we do this? And so with that, I think that's my last slide. I'm going to, just one summary. So I think we are seeing recent successes in clinical trials with monoclonal antibodies, which suggest removal of beta amyloid slows cognitive decline. It's a huge advance for the field. Additional therapeutic targets are under development, as well as future combination therapies. I think biomarkers are going to provide novel and accessible diagnostic, prognostic, and theragnostic tools, and will likely lead to precision medicine, which will be able to deliver the right treatments to the right patients. Huge challenges, but also opportunities for healthcare systems and the new treatment era. And health disparities and access to treatment for underrepresented populations really needs to be addressed. And finally, I'd sort of say, I hope I've convinced you that Indiana is really playing a central role and critical role in finding solutions for Alzheimer's disease patients, caregivers, and communities.
Thank you for getting through a huge amount of content in a very short amount of time. And I think I would challenge each and every one of you to find a discovery scientist anywhere else who can talk about the entire landscape from the earliest stage biomarkers and mouse models all the way to disparities and health system challenges. And I think it's a testament not just to Dr. Lamb, but also to the way our system works together in this community and how in Indiana we're not going to operate in silos. We may be right now, but we're going to pull ourselves out of them.